Good day everyone. In this presentation, I am going to discuss an overview about the cellular respiration, fermentation, and the step-by-step process of glycolysis. Okay, so when we say cellular respiration, it is a metabolic reaction that produces ATP or adenosine triphosphate with the presence of oxygen. In addition, cellular respiration is an exogenic process, and this process transfer energy is stored in the food molecules that we consume to produce ATP. And lastly, cellular respiration occurs or takes place in the cytoplasm of the cell and in the mitochondria of eukaryotic cell. Okay, so in cellular respiration, it involves series of reactions, and the main reaction involved is we have the redox reaction. Okay, so this redox reaction is a transfer of one or more electrons from one reactant to another. And the two types of redox reaction are oxidation and reduction. Okay, so in oxidation, it loses a electron from a substances and gains oxygen, while in reduction, it gains electrons from a substance or molecules and it loses oxygen molecule. Okay, so we have here the overall balance equation for cellular respiration. Okay, so C6H12O6 or glucose plus 6 molecule of oxygen will result to the production of 6 molecules of carbon dioxide, 6 molecules of water, and a total of 36 ATP or energy. Okay, so later on, after nating ma-discuss yung electron transport chain, we are going to solve for the unbalanced equation of this equation. Okay, so titignan natin kung paano ba nakuha tong overall equation na to. Okay, so cellular respiration involves three main metabolic stages. These are glycolysis. Okay, so glycolysis occurs in the cytoplasm of a eukaryotic and prokaryotic cell. Next is we have the Krebs cycle or the citric acid cycle. Okay, so this Krebs cycle occurs in the matrix of the mitochondria. And lastly, we have the electron transport chain or the oxidative phosphorylation. And this third stage occurs in the inner membrane of the mitochondria. Okay, so now let's discuss the first metabolic stage of cellular respiration, which is glycolysis. Okay, so glycolysis is a chemical reaction that releases or produces energy from a glucose compound. In addition, glycolysis that does not require the presence of oxygen, meaning it is an anaerobic. And glycolysis occurs very rapid in the cytoplasm of eukaryotic and prokaryotic cell. Okay, so for the overview of the glycolysis, okay, so glucose is a 6 carbon ring molecule that is found in the blood and is usually a result of breakdown of carbohydrates into sugar. Okay, so this in glycolysis, one molecule of glucose is broken down into 2 pyruvate. And pyruvate is a 3-carbon compound. And as a result, it produces ATP and NADPH, or what we call the nicotinamide adenine phosphate hydride. And this pyruvate is further metabolized in the energy pathway to produce more ATP. Okay, so glycolysis involves 9 distinct reactions. Okay, so it starts from the conversion of glucose into glucose 6-phosphate. Okay, so this conversion will be possible through the help of the enzyme hexokinase. Okay, so hexokinase or the enzyme hexokinase will transfer a molecule of phosphate from ATP to convert glucose into glucose 6-phosphate. And this process of addition of phosphate group to a molecule is known as a phosphorylation reaction. In this first step of glycolysis, ATP is converted into ADP. Hence, it utilizes one molecule of ATP. Okay, so the second step of glycolysis involves the conversion of glucose 6-phosphate to fructose 6-phosphate. And this reaction occurs with the help of the enzyme phosphoglucose isomerase. Okay, so as the name of the enzyme suggests, this reaction involves a isomerization reaction. Okay, so this reaction involves the rearrangement of the carbon-oxygen bond to transform the 6-membered ring into a 5-membered ring. And as a result, the glucose 6-phosphate is converted into fructose 6-phosphate. Okay, so in the third step of glycolysis, fructose 6-phosphate is converted into fructose 1,6 bisphosphate or FBP. Okay, so the enzyme that catalyzes this reaction is the phosphofructokinase. Okay, so similar to the reaction of the or occur in the step 1 of glycolysis, second molecule of ATP provides a phosphate group that is added to the phosphate fructose 6-phosphate. Okay, so the addition of phosphate group will result to fructose 1,6 bisphosphate. And again, phosphorylation reaction takes place and ATP is converted into ADP. Hence, another ATP is utilized in this reaction. Okay, so for the final step of the first stage of glycolysis, it is utilized by the enzyme aldolase. Okay, so these enzymes catalyzes the cleavage of fructose 1,6 bisphosphate and yields into a 2,3-carbon molecules. And these molecules are glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Okay, so GAP or the glyceraldehyde 3-phosphate is the only molecule that continues in the glycolytic pathway. Okay, so to continue the process of glycolysis, the dihydroxyacetone phosphate or the DAP molecules produced are further acted on by the enzyme triose phosphate isomerase or triphosphate isomerase. Okay, so this enzyme reorganizes the DAP or the dihydroxyacetone phosphate into a glyceraldehyde 3-phosphate to continue the process of glycolysis. Okay, so the first 5 step comprises the first phase of glycolysis, also known as the energy investment phase wherein 2 ATP is used in especially in the step 1 and 3 and 0 ATP is produced. And lastly, at this point of glycolytic pathway, we have 2 3-carbon molecules. Okay, so moving on for the 6th step. Okay, so glyceraldehyde 3-phosphate is oxidized by the coenzyme nicotinamide adenine dinucleotide or known as the NAD. Okay, so the enzymes that catalyzes this reaction is known as the glyceraldehyde 3-phosphate dehydrogenase or known as the GAPDH or GAPDH. Okay, so generally, the enzyme GAPDH or glyceraldehyde 3-phosphate dehydrogenase will allow the NAD molecule to pull a hydrogen molecule from the glyceraldehyde 3-phosphate. As a result, NAD is converted into NADH, or known as the nicotinamide adenine dinucleotide hydride. Okay, so the energy produced by the conversion of NAD to NADH is used to attach the inorganic phosphate to oxidize the product form or to yield 1,3-bisphosphoglycerate. And lastly, the NADH produced is further utilized in the electron transport chain to produce more energy. Okay, so in this step, the 1,3-bisphosphoglycerate is converted to 3-phosphoglycerate by the enzyme phosphoglycerate kinase. In this reaction, it involves the loss of phosphate group. Okay, so the phosphate group is transferred to a molecule of ADP that yields to our first molecule of ATP. For the next step, it involves a simple rearrangement of the position of phosphate group to a 3-phosphoglycerate molecule, making it 2-phosphoglycerate. And the enzyme that catalyzes this reaction is called or known as the phosphoglycerate mutase. Okay, so a mutase or an enzyme mutase is an enzyme that catalyzes a transfer of a functional group from a position of the molecule to another. Okay, so the reaction mechanism proceeds by the adding of additional phosphate group to the second position of the 3-phosphoglycerate. Okay, so the enzyme will remove the phosphate from the third position, leaving just 2-phosphate, and thus it yields a 2-phosphoglycerate. Okay, so the enolase enzyme work by removing a water group or dehydrating the 2-phosphoglycerate. Okay, so as a result of the dehydration of 2-phosphoglycerate, it will yield 2-phosphoenol pyruvate. Okay, so the final step of glycolysis involves the conversion of phosphoenol pyruvate into a pyruvate with the help of the enzyme pyruvate kinase. Okay, so in this reaction, a transfer of phosphate group is involved. Okay, so the phosphate group that is attached to the second carbon of the phosphoenol pyruvate is transferred to a molecule of ADP. Okay, so yielding another ATP. Okay, so again, since there are two molecules of PEP or the phosphoenol pyruvate, so we have here in this reaction we generated two ATP molecules. Okay, so the second phase of glycolysis comprises the 6 to 9 steps, and the second phase of glycolysis is also known as the energy yielding phase. Okay, so the result of second phase of glycolysis is our 4 molecule of ADP, or should I say 4 molecule of ADP is used, especially in the step 7 and 9. And there are 2 net gain of ATP produced and another 2 molecule of NADH. And lastly, we generated 2 molecules of pyruvate. Okay, so to sum it up, the result of glycolysis is 2 3-carbon pyruvate, 2 ATP, and 2 molecule of NADH. Okay, so take note that these 2 pyruvate are transported from the cytoplasm into the mitochondria of the cell, and it is converted into 2-acetyl-CoH, and it will be used for the second stage of cellular respiration, which is the Krebs cycle. Okay, so there are 2 fates of the pyruvate produced in glycolysis. Okay, so first, with the presence of oxygen, it will continue in the Krebs cycle and electron transport chain in the aerobic respiration. But without oxygen, it will proceed to fermentation. So either lactic acid fermentation or alcoholic fermentation depends on the organism.